Good afternoon, everybody. I'm Susanna B, and my supervisor is Dr. Ong Ming Tong, and my project is on the study on the anti-proliferation property of lavender essential oil using cell-based assay. My study involves MCF7, which is a breast cancer cell. So, a little intro on breast cancer. It is the most diagnosed cancer among women. It ranks second most cancer-related deaths in females globally, and people living in high index, high very high human development index and very low human development index are mostly diagnosed with breast cancer during their lifetime. Next, the um, the plant that the, the plant that I will be working with is the lavender essential oil. Lavender essential oil is traditionally used for medicines for centuries due to the therapeutic uses. So countries like China, Egypt, India, and Iran have been actively conducting research on lavender essential oil, particularly exploring its application in traditional medicine and aromatherapy. So the medical attributes have contributed widely for its popularity, for its therapeutic uses. So recent studies have emphasized the anxiolytic, anti-inflammatory, anti-nociceptive, antimicrobial, and antioxidant effects of lavender essential oil, suggesting its potential as alternatives to synthetic materials across different domains. So, my research hypothesis or research question is: will the commercially available lavender essential oil exhibit an anti proliferatory effect on MCF7 cells, which will be measured by MTT as an IC50 maximal inhibitory concentration? Next, the literature review. So, Ovedi et al. And the researchers identify different chemical composition of the lavender essential oil and also demonstrated the anti-proliferatory activity of the essential oil and various cancer cell lines. Next, Femi and researchers, they also demonstrated the compositions of the lavender essential oil and also showed the anti-cancer and anti-mutagenic effect of it on the HEP, G2, and E549 cell lines. Next, researchers they used different solvents for the lavender essential oil. They used ethanol, methanol, extract, and on that they found that ethanol extract of the lavender exhibited superior cytotoxicity and safety profiles against the breast cancer cell lines. They also found that in vivo study, there's a 43.29% reduction in tumor sizes, which was greater than other solvent extract that they used in the study. Next, researchers um, found the major components in the lavender essential oil and also demonstrated the antioxidant and anti-proliferatory effects of lavender essential oil across various cancer cell lines. Next, my research methodology is to use the commercially available, that is the Vanzo lavender essential oil, and figuring out the antioxidant activity, anti-proliferative activity, profiling the sample, and figuring out the anti-inflammatory activity of Vanzo commercial essential oil. So, for this progress report, I'm gonna elucidate on the antioxidant assay, the DPPH assay. So for DPPH, I made concentrations, different concentrations of samples, standard and DPPH. Next, I added them on the 96 well plate and incubated for 30 minutes in the dark. And after the 30 minutes, have read it on the spectrophotometer at solvents of wavelength 517 nanometers. Next, I plotted the graph for radical scavenging capacity in percentage. So this is the DPPH results. So here we can see the different concentration of lavender essential oil and the radical scavenging activity of it's increasing. So the highest percentage of activity is on 0.8, and on the right side, the standard that I used was ascorbic acid. So here's the radical scavenging activity of ascorbic acid. So the results demonstrate that the lavender essential oil exhibit antioxidant activity as compared to the standard ascorbic acid, while it did decrease the DPPH absorbance. Its ability was notably lower than that of ascorbic acid. This indicates that the sample has the capability to donate hydrogen atoms or electrons to neutralize the DPPH radical. It is less effective than ascorbic acid in doing so. So here is the combined result, the sample and the standard. In conclusion, I would like to say that TPPH results indicate that the oil do have antioxidant activity, as it cannot be compared to the standard, but it still does have the ability. So for future studies, the other antioxidant assays can be used, or combinations of other antioxidant can be used to elucidate its mechanism more, or yeah, different free radical assays can be used for comprehensive understanding. This is the gun chart. So far I'm still progressing as planned and will progress as planned, hopefully, with the references. Thank you.
Uh,
Dr. Eric, do you want to
Yeah.
ask?
yes, I can ask first. Sultana, can you go back to slide number 8?
Yeah.
This slide, one is the what sample? This one have the done in triplicate or what? No, right? I think so, right?
Sorry, I
Sample.
did not get you. The sample is the Vanzo commercial lavender essential oil.
Again? Sorry. Pardon?
It's the Vanzo lavender essential oil.
Oh, this sample is run in triplicate or not?
Yeah,
Or only one sample?
yeah, I did run it in duplicate.
But
Duplicate
there
or
is no,
triplicate?
there are no standard deviation or what here? SD. You have SD or not?
No, this doesn't, like, I did not calculate the standard deviation. I just calculated the percentage of radical scavenging activity.
Oh, this is percentage, yeah?
Yeah,
So you compare to the standard, so it show reduction or what? So compared to standard, it show this is antioxidant activity, right? So it's only about 20% of the standard.
yeah,
Yeah, the maximum it goes to
30%.
20%, right? Hmm. Previous slide. Can you back previous slide? This is what? This is the deprivation result also. This is you only have radical stability. This slide is similar to the slide number 8. No, different experiment or what?
It's the same.
It's the same, right?
Yeah.
I don't know lah because so the conclusion is really 20% out of the standard. You mean your sample lah, what you expected. So it's good or bad? I don't know.
Compared to the standard, it's not up to the standard, but it is showing an antioxidant activity.
So, but what the point, right? Actually, right? So you only about 20%. Why they don't use the standard? So I think you have to identify what the benefit. Not only show that you have show the antioxidant activity. People ask, you only show 20% out of the standard. Why they want to use it? Any benefit other than other thing? What else experiment you gonna do for this activity? Can you go back again to the previous slide? Others, what other experiment? Antioxidant. What else you gonna do? Can you tell us what else?
Research methodology.
Oh, okay, this one, right? So antioxidant only show about 20%. Yes, it show activity, but I would consider it's quite low lah when you compare the standard. Sure. Anti-privation. So you only complete the antioxidant right now so far.
Yeah.
Yeah. I cannot comment too much about it, but because I don't know what the others. I think the main one will be anti-inflammatory and anti-proliferation. I don't know
Yeah.
what to. Oh, yeah. So maybe antioxidant just by taste for it. Not so important. So I cannot comment too much. But you have to think lah. It's only about 20%. It's quite low. So you have the other justification. Maybe like I say higher activity in anti-proliferation or anti-inflammatory. Okay. Overall, it's good for progress. That's all for me. Dr. Koo.
Okay, doctor. Thank you.
So Tana, can you go back to the results? Second slide, sorry, of the result.
This one?
Actually, I don't know also what to ask. Second.
Okay.
This is standard curve. Just now, Dr. Eric asked how many replications?
Yeah, I ran the
Any
experiment in duplicates.
duplicate,
Triplicate. Yeah.
triplicate.
Yeah.
And then how many biological replications? Triplicate is technical replications. How many biological replications? Meaning a same sample repeat 3 times. This is replications. And then different samples. How many times have you repeated?
Oh, I just
If-
repeated one time with 3 sets.
You may repeat again. Try to repeat again. See whether the result is consistent. Consistent at 20%. 20% right now, see again, not more than 20%. Not more than 30%, less than 30%.
Yeah.
Can you repeat again using different samples?
Different, you mean different extract or different essential oil?
Different extracts, different extract.
Oh.
Can you understand? Still your
Um,
essential oil. Is that correct? You are studying on
Yeah.
essential oil. Yeah. The essential oil you extracted from different time.
No, I didn't extract it. I got it.
You bought,
Yeah. It's a commercially
then
available.
different experimental environment. I think you need at least twice. And then every time you prepare triplicate, then we can see your the value of your experiment. Right now we cannot see because just one dot. You don't have the standard deviations or standard error mean. Can you
Oh, for
understand?
this, yeah. But for this experiment, I've seen the researchers have just took the
Yeah, just one line.
percentage. Yeah, they just took the
Meaning there is no
percentage.
statistical analysis.
Okay.
You may consider,
Okay. Okay, doctor.
but your supervisor should agree with this. Can you go back to the Gantt chart? So where are you now?
I'm figuring out the levels of ethanol that will potentially kill the cells. Like I'm figuring out the concentration of ethanol to carry out the MTT
So
with
you have
essential.
spent 1.5 months for the antioxidant assay.
Yeah.
You need another 2 months for the for the cell viability test.
Yeah. Hopefully.
Hopefully, okay. Right. Can I see your title again? Because you have 4 things: antioxidants, anti-proliferations, phytochemical tests, anti-inflammations. But why your title is only on anti-proliferations? Can I ask? Can I ask your the can I see the title again?
So it's like
Anti-property,
the yeah.
anti-proliferations.
It's like I'm focusing on the anti-cancer property of the lavender essential oil, but also I want to know the antioxidant and other aspects of the oil.
So what do you expect from your anti-proliferative assay when the antioxidant activity is less than 30%?
I don't know. I cannot predict it.
You expect. What do you expect? Your hypothesis. No, huh? Okay.
I think there will be some activity,
Effect.
little bit.
Are you sure?
I'm hoping.
Okay. Right.
Yeah,
Thank you, Sultana.
yeah. Thank you, doctor.
Bob, habis
Okay, habis. Thank you, doktor.
Ada soalan lagi ya, Dr. Eric?
Hmm. Tadi ada apa-apa lagi ke? Tanya?
Tak ada.
Okay. Thank you.
Thank you.
Bob nak share session ni juga atau saya share?
Untuk yang postpone ni, Dr. Chu Island dengan Sida tak masuk lagi. Mungkin akan masuk 245 tu kalau ikut time.
Oh, Dr. Jiwa Island yang share kan?
Tak apa, saya boleh share, doktor.
Okay. Thank you.
We are waiting for who?
We're waiting for the Presiden at the Island because the schedule to 45.
Oh, okay.
Can wait 5 minutes, Dr. Chu Island turning the PC.
Bob,
Betul?
ataupun kita start dengan yang selepas tapa tu.
Buat apa tu? Data UG tak ada lagi.
Oh ya ke.
Data pertambangan ni tak masuk lagi. Sebab data C dia tengah buka bentar tu.
Okay.
Hello. Maybe we can start now because Dr. Chu has problem with the WebEx. Puspa, can we start now?
Hi. Yes.
Can we start now?
Yeah. Is my voice audible?
Yeah. Let's start. The check.
Good afternoon. Can you see my slide?
Sorry, I'm here. Hello.
Doctor,
Yeah,
can you
yeah,
see-
okay, doctor.
Can you see my slide?
Hello. Faiz.
Yeah.
Okay.
Boleh start. Okay, doctor.
Sorry for delay. Sorry.
Hello. Hello.
Yeah, Puspa, you may start now. Just
Yes,
put your presentation to the presentation mode.
You
Thank
can see
you.
my slide.
Can see you. Yeah, okay, good.
Good afternoon, doctors and everyone else. I am Puspa, and today I'm going to present on the progress of my research, which is entitled Evaluation of Antimicrobial Activity of Medicinal Plant-Based Green Carbon Dots against Pathogenic Microbes, and my supervisor is Dr. Sasi. The presentation contains introduction, hypothesis, literature review, methods, findings, conclusion, and Gantt chart. Introduction. Antimicrobial resistance, aka AMR, it is a global health threat at the moment. It is caused due to the inappropriate use of antibiotics in the hospital settings and in the agriculture. Microorganisms, they develop resistance against the antibiotics due to the mutation, or they acquire resistance genes from the other microbes. So in 2019, WHO, they reported that about 1.27 million deaths occur annually due to antimicrobial resistance, and this number is likely to increase to 10 million by 2050 if nothing is, or if no action is taken against antibacterial resistant strains. So therefore, there is an urgent need to develop new antibiotics for these resistant bacterial strains. The development of nanotechnology, it has provided a promising field for developing new antimicrobial agents. Among various nanomaterials available, green carbon dots have gained a lot of attention from the researchers lately because of their unique physiochemical properties such as biocompatibility, solubility, tunability, stability, and antimicrobial activity. Carbon dots, these are like quasi-spherical, which is neither spherical nor a circle. They are zero-dimensional and their size is 10 nanometer or less. Green carbon dots, they are synthesized from natural resources. So they are environmental friendly, cost effective, and sustainable. Green carbon dots, they have shown remarkable potential in various applications such as biosensing, DNA sensing, bioimaging, and many others. The unique properties such as the small size and the presence of functional group on the surface of carbon dots have, they enable these particles to interact with the cell membrane of the microbes and then help them to like disrupt the cell membranes and then also to interfere with their internal processes. In this research, I used Polyalcia longifolia. It is an evergreen tree or medicinal plant which is mostly found in the subtropical regions such as Malaysia, Sri Lanka, India, Myanmar, etc. The leaves of this, I'll be using the leaves of this tree. So the leaves contains various compounds such as alkaloids, flavonoids, tannins, terpenes, etc. And this plant has also been used traditionally for the treatment of diseases such as body pain, gastrointestinal ulcers, skin diseases, fever, etc. So the hypothesis is that the synthesis of carbon dots from the leaves of P. longifolia will result in efficient and reproducible methods for the production of antibacterial nanoparticles. The characterization of synthesized carbon dots will reveal distinct structural and chemical properties that will be suitable for antibacterial application, and it will exhibit antibacterial activity against the targeted bacterial strains. Literature review. So in the past also, the other researchers, they have used these natural resources to synthesize carbon dots, and they have demonstrated the antibacterial activity. For example, Jeda, they also synthesize carbon dots from neem leaves, and so synthesized carbon dots, it also exhibited antimicrobial activity against bacteria and fungal strains. Another study conducted by Saravana, they also synthesized carbon dots using turmeric leaves, and it also had effective antimicrobial property against both gram-negative and gram-positive. A review article which was written by Narmin Mamin Hussain, they also discussed about the antimicrobial application of carbon dots and also highlighted their different properties such as low toxicity, ease of synthesis, and biocompatibility. Methods and data collection. For the synthesis of carbon dots, fresh P. longifolia leaves were first collected, washed thoroughly with distilled water, and then to remove any impurities, and then air dried. The dried leaves were then cut into small pieces, and then once the pieces were cut, it was subjected to pyrolysis at 300 degrees Celsius for 2 hours. Then the obtained brownish color residue was then grounded to powder using mortar and pestle, and then the about 10 mg of this powder was taken and dissolved in 100 ml of distilled water, and then tonicated, centrifuged, filtered, and the filtered was then sent for the freeze drying. So after freeze drying, the carbon dots were obtained in the powder form. So after obtaining a powdered form of carbon dot, it was characterized. The characterization was done using different techniques such as the potential, UV-Vis, FTIR, and TEM. Zeta potential, it was done to determine the surface charge and the stability of the carbon dots, UV-Vis to see its optical properties, and FTIR to determine different functional groups which is present on the surface of the carbon dots, and then transmission electron microscope to it was carried out to study the shape and size of the carbon dots. So after the characterization, antimicrobial assessment of the carbon dots will be done. So for the antimicrobial assessment, first the bacterial culture will be prepared. Bacterial subtilis will be cultured in laurea broth and then incubated at 37 degrees Celsius overnight in a shaker incubator. And then its OD will be measured. Then a bacterial suspension of OD 0.1 will be made, and then it will be used to carry out the MIC using broth dilution. And then after the assessment of antimicrobial activity to study the impact of carbon dots on the bacterial cell, it will be further subjected to a scanning electron microscope. So the findings: synthesis of carbon dots. After obtaining carbon dots in powder form from dry freezing, a small amount of it was taken and then dissolved in water. And it completely dissolved in water and gave a light yellowish color. So it indicates that they disperse while without any aggregation. Then it was taken and then observed under the UV 365 nanometer. The carbon dot exhibited greenish fluorescent. The exhibition of green color suggests that the synthesized carbon dots have specific size distribution and surface state that emit in the green region of the spectrum when excited by UV 365 nanometer. Characterizations of the carbon dots, zeta potential. This is a graph of zeta potential distribution. The x-axis represents apparent zeta potential, and y-axis represents total counts, which corresponds to the number of particles that is being measured at each zeta potential value. The distribution appears narrow and centered. So narrow and centered indicates that the particles have similar surface charge, which is good for the stability. The distribution, it has a peak, which the peak is observed at approximately minus 28.4 MV. So this indicates that the electrostatic repulsion between the particles in the carbon dot is consistent. So this means there will be no aggregations, and they will not form fluctuations or agglutinations. This is the spectrum of FTIR. It reveals the FTIR is carried out to find out the different functional groups present in the carbon dots. The different peaks here represents different functional groups. Example, hydroxyl group, CH trace. So this the peaks, the sharp peaks, they indicate it indicates the concentration of the particular functional group. For example, this peak, which is at 85.26, this represents that this is a peak for the aromatic group, and then this sharp point it indicates that there is mostly this aromatic group. For example, compared to this broad kind of peak here, it means that there are, apart from the OH hydroxyl group, there are other compounds also. And then in this part, here, 5 to 5, this peaks here. This, since it's like 525.55 and 608.83, these peaks are not actually the functional group, but what this could represent is the formation of compounds with the interaction of atoms with each other, and not actually a functional group. This is a graph for the UV spectrum of carbon dots. X-axis represents the wavelength and y-axis represents the absorption. So there is a peak. There's a small peak here and here between 2008 and 300 nanometer. And the spectrum starting 300, it starts to decline as the wavelength increases to 600 to 600 or 700. So this is a common feature or properties in the carbon dots, which indicates that the presence of transitions associated with various surface functional group. This is the histogram of TEM. Its analysis is still not yet completed. So milestones achieved. So far, the synthesis of carbon dot has been completed, and its characterization, UVVs, zeta potential, and FTIR has been completed. The remaining parts are TEM imaging, its analysis, and the antibacterial activity is yet to be completed. So the conclusion, so far the project is progressing well in accomplishing its initial objectives, and as outlined in the proposal, and aims to complete the projects within the remaining activities within the. Thank you. This is my one shot. So far, I've completed till here, but like some of the analysis part is still left, and this part are yet to be conducted. And these are the references. Thank you.
Thank you, Pushpa. Now I pass the time to evaluators. Dr. Shaida or Dr. Chiu, who would like to start? Right?
Can I ask question first?
Sure, please.
Thank you. Okay. Hi, Pushpa.
Dr.
Good presentation, and your achievement also is good. It's just my concern. Now, when you do the fluorescence checking on the carbon dot substance, and you compare with water, right? Do you have any other compounds that you can compare to show that only your carbon dots have a fluorescent, and the other, like a negative control other than water?
You mean for the fluorescence part?
Yeah.
Apparently, I don't think. I mean, there are other substances like rhodamine, but I'm not sure if we have that in the lab.
Because my concern is your analysis is based on characterizing the carbon dots, like you do FTIR, you do zeta potential, but is there any way that you can confirm, validate that this is carbon dots other than looking at the fluorescence ability?
Like it's TEM, like looking at the TEM, we can, like, depending on its size.
Depending on the size. Okay,
Yeah.
what about the others reported in the publication? Are they getting the same results like when you do zeta potential, you look at the minus 28, right? MV. Are they having the same results?
Yes. So in some of the research papers, they did have this zeta potential of not exactly minus 28, but like
In the range, the same range.
yeah, in the same range. For example, even one of the student who was doing the kind of similar research as me in the lab, she also has similar range for the zeta potential. So I think that kind of correlates.
Maybe you can maybe in future when you present you can put what are the findings from others, like this is according to other study, corroborated with other study that they found the same quite similar thing, just to confirm that you are extracting, getting the right carbon dots. I guess your analysis on bacteria antibacterial would not validate that your carbon dots are working, Is-
Another experiment that I can do is quantum yield. So quantum yield, it kind of quantifies the fluorescence of the carbon dot. So I can do that, and if the quantum yield is like around 8 or 7, it's a good carbon dot. But
You mean
if
at certain wavelength?
I mean, it's just so far I just know the 350 wavelength.
Wavelength.
Yeah.
As long as it can validate that you're getting the right carbon dots. It's a proven carbon dots. And then how are you gonna do the assessment for antibacterial activity?
But for that I'll be using broth microdilution method. So I'll be doing serial dilution. The highest concentration will be 1000 microliter per ml.
So if it can kill the bacteria, it means that it has the carbon dots, the potential lah.
It has the potential to kill the bacteria.
Kill bacteria. Okay, then we'll wait for that. That's all from me, Dr. Ong.
Hello, Puspa.
Yeah, thank you, Dr. Shaida. Yeah. Dr. Chiu, please go ahead.
Yeah, Puspa. Going back to your proposal that day, right? May I know what is the yield you have done your work on the preparation of carbon dots, right? So, how much roughly is the yield of your carbon dots?
Sorry, doctor, can you repeat the question, please?
The yield of your carbon dots.
Oh, I haven't actually done that proper way. Recently, I just kind of did a test kind of thing, so I haven't been able to do the calculations.
Okay. One more thing is, may you explain what elements of the carbon dots that give out the fluorescent under UV 365 nanometer?
Sorry, doctor.
Yeah, what is the possible elements that make it fluorescent under UV 365?
So far, my carbon dots and the research I've done, I found that aromatic like if there's a functional group aromatic, then they give this fluorescence.
So meaning there's something at 365 nanometer, right? My question is, because your UVVs, there's no peak at 365. Are they related? These two, are they related?
Yes, it's not compulsory that they have to produce the this absorption thing at 350. It can go way down below 300. So not all carbon dots they have the same ability to absorb the light. So in some of the this absorption of the light, it also depends on how you synthesize the carbon dots. So different methods of synthesizing, it leads to the formation of different sort of carbon dots. So it does not produce the same type of carbon dots, and depending on this, and also the presence of functional groups. So this all kind of plays role in the absorbance, in what wavelength they absorb the light.
Yeah. Please excuse my ignorance on this matter because I am not expert on carbon dots. I'm just curious because you say there is something like under 365. So I normally you expect like if then at UV 365, right? I would expect to see a peak or something there, right? Because that's the characteristics of a carbon dots. So I expect when you scan your sample, then we should expect something, a peak or something at 365. I mean, normal based on, I don't know, my understanding, I don't know whether it's correct or not, but I just wanted.
Okay. This
Do you understand
the
my
UVV's
questions?
spectrum. For the
Yeah.
UVV's, for the UVV's spectrum, like I said, they don't have the exact wavelength. So the graph that I have presented here is not, it's not what others have got in their research project. So for me, I have to kind of I might have done some errors while doing this experiment. So I think I might have to repeat this to to do it again and to make sure that it's actually kind of similar to the other research papers.
Okay. And one more thing is regarding that day, I think Dr. Ong did offer you some microbes, right, to work on the antimicrobial assay, right? You propose only one bacteria, right, that day?
Yes.
Right?
Yes.
In the initial proposal presentation, you proposed only one bacteria for your antimicrobial assay, right? If I am not, if I get it right.
Yeah. About that, since I haven't yet started for the antimicrobial thing, like at the moment I am kind of preparing the materials and required materials. So I need to go and speak to my supervisor, and I have to see what he wants me to kind of do.
So any progress on that direction? I mean, do you manage to decide or get more microbes rather than only one?
I did manage to get one fungal strain and one S virus.
Okay. One more thing that I noticed is that your title, right, is still like one of medicinal plants. So maybe in future, maybe you could discuss with supervisor, why not to change it to polyfolia, longifolia, right? P. longifolia, instead of medicinal plant to be more specific for the title. Because you are working with P. longifolia, right? Instead of putting a medicinal plant, it will be too general, I think. Okay.
Okay, doctor.
Yeah. I think that's all from me for today. Thank you.
Thank you, doctor.
Okay, thank you, Dr. Chiu. Pushpa, when you answer the question on on the question that Dr. Chu asked you, absorbance at 300 something. I think that you need to go ahead with the explanation by looking at how the color you can see the color of your carbon dot suspension. So from there you might be able to answer Dr. Chiu. Okay, so go and have a look at from that perspective, and if you can't understand, you may ask your supervisor. All right. Okay. Thank you, Pushpa. So,
Thank you, Doctor.
all right. Thank you. Dr. Sasi is not around, right? So maybe I just go ahead with the next presentation. So next presenter will be Liu Yap Si Jian. His supervisor is Dr. Eugene. Evaluators Dr. Fatma and Dr. Nick Hughes. All right. Liu, are you ready?
Yes.
Okay. Dr. Fatma is around, and Dr. Nick Hughes, is she around?
Tak nampak pula. Dr.
Tak
Nick
ada
Hughes in
lagi
the
ya.
list.
So we
Um,
need to wait a while
nanti
for her.
I panggil dia kejap. Can I call her?
Boleh,
Okay?
boleh. Ya, Dr. Fatma. Thank you.
Okay.
Yeah.
Hello, hi.
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 Yeah, Dr. Nick Hughes is here. Okay. So everybody is around. So let's start now. Liu, you may continue. You may start now. Thank you.
Okay. Good afternoon to Dr. Fatma and Dr. Nick, and also my supervisor, Dr. Eugene. So I'm Leo. Today I'm going to present my progress based on my progress on my dissertations. So again, my title is machine learning based prediction method in identifying skin cancer type by microRNA expression profile. Okay. So introduction. Just a brief one. Skin cancer is characterized by abnormal growth of skin cells due to overexposure to UV ray from the sun, tanning beds, activities. So this will cause the DNA mutation, triggering uncontrolled proliferation of skin cells. So in general, skin cancer can be caused by two cancer cells. Okay. So one is melanoma and one is a non-melanoma. So melanoma specifically is refers to the skin cancer cell, the tumor developed by the melanocytes, while non-melanoma is the cells that is otherwise from the melanocyte. So the most common one could be basal cell carcinoma and squamous cell carcinoma. Okay, so moving on to problem statement. So the reason why we raise this idea is because conventional methods of skin cancer diagnosis often rely on visual inspections, so which can be subjective and prone to error. So the hypothesis, so the problem statement is the machine learning is able to provide prediction of skin cancer type based on microRNA expression profiles, and the hypothesis is by these two. So it's original from the proposal. Okay, it's still the same. So literature review, okay, of course, the main things here is the microRNA and machine learning. So microRNA are short non-coding single-stranded RNA molecules, which play a crucial role in oncogenic activities by managing mRNA expressions. While on the other hand, machine learning is a method to train computers by developing algorithms to serve the purpose of identifying patterns, or in this case, is making predictions. Okay, so for now, the progress, I would say, I finished the data mining part. Okay, still yet to start on the data cleaning part. So the method and data collection and the finding there later will be mainly focused on the data found. So okay, so the primary data sources is from this website. It's called the Cancer Genome Atlas Program, TCGA. So it's databases which consists of a lot of different sequencing profile of cancers. So you may find like exome sequencing, not like RNA sequencing there. There's a lot of like this kind of data there. So the primary source is from here. So how do we get the data from there is that this is the basic workflow. So you visit it, and then you may need to download the transfer data tools, and then you download the manifest, and then the data tools will convert the manifest, which is contain the informations of the things that you want to download, and then it will convert into data. So the other sources is from this gene expression omnibus. I'll discuss later why I use this. I am not proposed this originally in my proposal. Okay, so an ideal data that can be found from the TCGA is somewhat the data looks similar like this. So which you have the mRNA name, and then you have the read count. So this is the microRNA expression profile by sequencing. So that indicate these read counts, and then with also the mRNA names. So other than that, where's my, I'm gonna show another thing. Yeah, here. So other than that, after I got this sort of the data, the mRNA expression profile data, and then I also need to label all this and I label all this based on the disease type and its UUID. UUID is something like its identity, on that data, and then label it based on the disease type. So this is the very ideal conditions of the data. However, after searching after data mining on the TCGA, the thing is the data I found there is mainly the melanoma data only. They don't have any non-melanoma cancer cell data. So this leads to the question on how do I get this. So I have to go to online and search for papers. Otherwise I couldn't do anything about it. So that's why I use this gene expression omnibus. It's also part of NCBI for you to access to the others microRNA data on internet. So it's a bit non-ideal because the reason why is the data mostly presented from there is something look like this, which is not a microRNA expression profile, but the RT-qPCR data. So which is a bit troublesome, okay? But this is already been, I'm not sure it's already been considered. Yeah, it's already been considered previously on proposal. So yeah, this is all non-ideal data. So moving to a conclusion, the process of data mining is completed. So moving on to the next stage, integrating data that is from the RT-qPCR and the microRNA expression profile will be the next challenge. So this is my Gantt chart. Okay. So so I think that's all from me today. So yeah, thank you.
Thank you, Leo. Dr. Padma, would you like to start?
Thank you, Dr. Ong. My internet is a bit slow. But you guys You can hear me, right?
Yeah, I
Yeah,
can
very,
hear you and
very
see you
clear.
clearly. Yeah.
Okay. Okay. So, Leo,
Yeah,
thank you very much for your presentation. I think I remember when you presented your proposal last time, it was a very not very while ago, kan? And then now you have your progress presentation.
yeah, it is.
When was it, the proposal?
It was, I would say, a month ago, roughly a month ago.
A month, a month ago,
Yeah,
But anyway, quite quite early kan, Dr. Ong? Is it? Macam ni eh?
yeah, it is.
Ya, cepat sangat lah rasanya.
Ah,
Because the designated date for the proposal was on May, the first week of May, if I'm not mistaken, and then
postpone
they postponed it. Yeah.
because of that lah.
Eh,
Okay. Okay, now, your presentation is, well, thankfully, your proposal was very not so long ago. So I can still recall lah what you have presented last time and then relate to what you have presented today.
Yes.
That is one thing, the positive side of this very early progress presentation. So now you have two cohorts for your machine learning, kan? You have your melanoma and non-melanoma.
Yeah.
But earlier this was not
Mentioned in the proposal.
mentioned in the proposal, kan? So now after discussion you have decided to choose melanoma and non-melanoma, yeah?
No, it was mentioned, but the
Sorry,
data,
sorry. The cohorts. The
yeah,
cohorts.
yeah.
Now, and then and now when you in your conclusion
Yeah.
you mentioned that the data sets are now completed.
The data mining process. I believe I
Data
got enough
mining.
data, but if it's not enough, I have to buy another one. This advice I
Okay.
have to ask from the company side, like the external people side. Ask for their advice,
Yeah.
see whether it's enough or
Yeah.
not.
Yeah. So when you say data mining is completed, you have identified the data sets that you want to use, right?
Yes.
So how many data sets? Because
Roughly,
you
I've not
presented
been.
here quite quick, so I did not.
Oh, it was not. I didn't calculated it, but it was a few hundreds. I think it's like 500 or 600 of data sets.
Data sets of the microRNA or the data sets of from the database?
Oh, data set for the microRNA. And then from the TCGA database is roughly around 400 something.
So when you say data sets here,
Ah,
is the microRNA.
yeah.
It's not the data sets from the database, because data sets from a database, they have a different call name, kan?
Yeah, I think so.
And then you don't have that. You don't put them as a results.
Temporarily not lah, because it's because all the data is like raw data and it's just together with, if all is on the
But
TXT file right now.
but in the data sets, in the call name of the data sets, you should have the detail of okay, so this data set is, you mentioned microarray and the other one is real time.
One is from one is from the
So one is
Luntine.
from so
One is
how
from
many from microarray and how many from real time PCR?
So yeah, from the micro array part is like 400 something, if I'm not.
That is that is the microRNA.
Yeah,
I mean,
it is the like 400 something is from the micro RNA sequencing
okay.
profile, and then the rest is from this database gene expression omnibus, because on this database itself it doesn't have the other one, the non-melanoma one. So that is the problem right now.
So what is the call name of the data sets?
I haven't. I'm not sure. I'm not sure about the name because I never considered about the name.
So Who did the data mining? You did yourself, kan?
Yeah.
So in the data sets, it should mention what is the control being used, the patient are coming from, how many are Caucasian, how many Hispanic.
Oh, I still
Yeah.
need to make statistics on that lah, but I already have the information together with the wait it's on the line. So you want me to show you, doctor?
Name and with the detail of from the every data sets that you get. When I said data sets, it's not the microRNA itself. That's your data. That's the raw data. What I mean is the detail of the data sets that you get because they have a call name. In a simple way, they usually have a gene number. Kalau it's a gene bank, kan, they have a gene number.
Oh,
So for data
yeah.
sets, they have a call for that data set, certain number, and then the experiment inside that call number, how many samples, what is the control, how many. So you don't show that in today, even though you say in conclusion, completed data mining, but I don't see that. So
Hello,
this
sorry.
is from,
Sorry.
yeah. So what
Yeah.
are you showing now?
Are you referring to this?
Yes.
That they
So
have
this is
ID.
the entity ID. So this is entity ID. This is coming from, this is for microRNA sahaja, yeah? MicroRNA seq.
Yeah.
So platform not mentioned. And then how, so this is, I suggest, I guess this is next gene sequencing, and microRNA seq. Yeah. So these are the details. How many of these kind of data sets that you use to get all the hundreds and hundreds of raw data?
Oh, it's all on here. This is all the data I used.
Okay. So how many are there?
452.
452.
Yeah.
$452 files, and how are you going to explain this in a results findings form? So you have 452 data sets containing probably a few thousands of raw data of microRNA.
Okay. So I should go and label all this and make a spreadsheet about that? You mean? Present all this in the presentation, right?
Yes. Yes.
Oh, okay.
Yeah. Because you are doing machine learning, and you have a big data sets of a few hundreds. I mean, in my case, we use like 12 data sets containing a few thousands of raw data, but you have 452, which is like what? 100 times 400, or maybe 100 times more than what I did with my students. So you should at least put this. So what I mean is your result should be structured lah. You cannot just say that, okay, this is the raw data, and then you completed your data mining. But the way it's just I cannot see the spell out of the results. You need to put the detail of your result. It should be in a structured manner, how many data sets you're using. And then when you say data mining is completed, there must be some redundancies, kan, when you
Yeah, it is.
compare between data sets. Have you compared between each data sets 452 redundant data in for number one compared to data from 452 microRNA, let's say microRNA 5 to 1. Is it different? Are they different?
I've taken a glance and some lah. I'm not sure about the others among all these 452s. But
Yeah, I mean,
so far what I can see is no redundancy yet.
so for each file, the microRNA are different in it from each. They don't quantify the same microRNA in the same file.
I would say as far as I see, it's not yet, but maybe there could be some inside. Because I haven't checked gone through all the this data yet.
So usually when you want to see some redundancy, you do a, what do you call that, a Venn diagram, kan? But I know you are working towards machine learning, but at least all this data of redundancy from each file, you should structurally put it in a visualized form so that we as a reviewer can see it. If you just spell out like that, I don't know how to assess. How do I say? I don't know how to assess it because your presentation is quite
Short.
simple. Yeah,
Yeah.
quite short and quite simple, and there's no structured result there for me to assess. So yeah, I think that's my most comment lah for this one. I pass to Dr. Nick lah.
Understood.
Mm.
I think I should do more about the present about the data.
Yeah,
Yeah,
it should have a structured presentation of the results. You cannot just simply say complete the data mining and then you show all the microRNA type, which is not all of it. You just a bit. Which I as a assessor I cannot say anything lah. I'm like, okay, well done, you know.
all right.
Which is not fair lah. I'm sure you have done something here. So you
Yeah.
need to fairly present it so that we can assess it fairly as well. But then
Okay.
it pass to you.
Okay, thank you, Dr. Fatma. Leo, I think you have to seriously take note of Dr. Fatma's comment because for me personally, I have the same feeling like Dr. Fatma. I see no structure presentation, and I don't know where to go. It seems that I just have to take for granted. Take your word.
Yeah,
What you
as
have
if
done, right?
you are
Yeah.
forcing us to say,
Yeah.
okay, you are good, you are on track.
Yeah. Okay.
Yeah.
So
Thanks.
let's see. Dr. Nick Hughes. Dr. Nick, please go ahead. Yeah.
Yeah, hi, Dr. Ong. Hi. Hi, everyone. Okay, Leo. It's very, very brief report on your result and progress. Perhaps maybe you can show. This is what I'm suggesting whether you can show to the audience how do you search for your data. Maybe perhaps what keywords that you use. You use TCGA, right?
Okay. Yeah.
TCGA.
So the keyword
what
I
keywords
use.
did you use?
You would like me to demonstrate now?
Not demonstrate, or I don't know. How about others? Would you like to see Leo to demonstrate, or just mention what keywords that you use on what database that you search for your data set?
It was also quite simple. It was just like clicking on filtering. So first of all, you go to this website, GDC data portal. So you probably go to the home page. So the next thing you want to find is the repository, and then you can click on this case here, and then you can make a filter on here. So there's like, let's say today there's skins, so I click on skins. So after that, I close this filter and then I will search on the experimental strategy to find the, so today, let's say I want microRNA sequencing. I'll just click this. Then on the data category, I just click on the transcriptome profiling. And then the data type, I click on microRNA expression quantifications. And yeah, I got the data here. But to extract the data from here, I have to use Linux because I found on the window is not working. So yeah, that's how I got my data.
So perhaps this kind of step you can also include on today's presentation, some things that you can present, because you don't have too much information to present. This kind of searching of data